Hello students, as we know, we are studying the cell signaling and this is the fourth class. Here we'll study about two parts that is secondary messenger and desensitization. As previous class, we have studied about GPCR. Most topic is related to that, signaling that based on that receptors and further mechanism or before reaching to the receptors. At first, what is cell signaling? As you know from the first class, I have been giving some information on that. The biochemical process where a cell responds to extracellular signals and act based on that signal or further rely the signal to the exact target site. In this signaling, two different messenger are involved, that is primary messenger and secondary messenger. Primary messenger are messenger that are normally as known as ligands. The information come from different cells and act on target cell through the primary messenger, that is the ligands like hormone, neurotransmitters, etc. These are the primary messenger. What are the secondary messenger? The messenger that are activated after the receptor receives the signals and stimulates other helper signals, the secondary messengers that are used for relaying the message from receptor to the target cell. That are called secondary messengers. For some, as for some example, CMP, IP3, some things, etc. Adenyl cyclase, more are there. More examples are there. You can study that's all. Let's talk about calcium. What is calcium and how it's act on the cell? On cell, it is act as resting cytosol concentration, where it kept extremely low to maintain a steep gradient compared to the extracellular fluid. It maintained uh, ion concentration from outer surface to inner surface. That is the intercell as other things each intracellular release, where the calcium released from the endoplasmic reticulum, where as where IP3 pathway act and activate the calcium receptor or calcium channel in the endoplasmic reticulum. At third, extracellular influx. Extracellular influx, here the cell outer environment and inner environment been linked with the channel, the voltage gated or ligand gated calcium channel through which the calcium can enter or exit. Through that, the ion has been balanced in the cell as per the outer surface or environment. What is the biological role? Calcium bind to specific sensor proteins like calmodulin, activate downstream kinase to trigger muscle contraction, neurotransmitter secretion, wildly expressed enzyme activations. These are when activated. Many more things are also been held, but importantly, these are the main mechanisms where calcium released from endoplasmic reticulums or came inside through the channel from outer environment to the inner environment. They are been held or bind together to form calmodulin. That calmodulin help in other functions such as contractions, neurotransmitter secretions, etc. These are the basically main uses or works of calcium in the cell. Next, the secondary messenger family. Here, Here, the small non-protein intercellular signaling molecules that minute size allow for rapid diffusion across the cell. This small secondary messenger, the small secondary messenger, are fastly diffused from mm, uh, are fastly diffused in the cells, and due to that, they can transport the message so fastly or speedily. What is the regulation? Continuous generated upon stimulation and rapid destroyed. They are generated fastly and destroyed also fast. Though there will be no continuous signaling due to that. If the detection does not occur, there will be the uh, miscalculations or missignal productions where um, if the signal is not disrupted, then it will show the ligand that receive or the target site will be continuously activate, and that will be miss messengers, or uh, there will be some problem of continuous activations of that work that can cause heart disease, etc. Common messengers that are the common secondary messengers that are cyclic AMP, cyclic GMP, IP3, DAG, calcium. How they are secreted? As we have shown before, ligands bind to the receptor, as shown here, GPCR receptor. GPCR activate G alpha S, G alpha Q, G alpha I, etc. And due to that, adenyl cyclase activate or PLC activate. When adenyl cyclase activate, cyclic AMP activate, and it is a secondary messenger. When PLC is activated, it cleave the PIP2 and form IP3 and DAG. And this IP3 and DAG are also the secondary messengers. There are two type of messengers, that is no, that one not. These are hydrophilic secondary messenger. What are hydrophilic? These are the polar molecules that can't enter the cell bilayer, plasma membrane layer, due to the, as you know further, that this plasma membrane or lipid bilayer is a hydrophobic uh, layer. Due to that, the hydrophilic substance cannot enter the hydrophobic layer. Due to that, the hydrophilic secondary messengers uh, will be bind in the receptor that are present in the plasma membrane, and through that the primary messengers activate bind to the receptors and activate the secondary messengers. How the secondary messengers navigate in cytosol? There are different signals that activate in different part, as I have shown in previous page, that the CAMP activate in the nucleus or other things, and the IP3, PIP3, that would be cleave, and IP3 would be product. That IP3 will be acting ER and release calcium. As written here, CAMP rapidly diffuse to bind to the regulatory subunit of protein kinase A, unleash catalytic uh, subunit to phosphorylate target, and the IP3 cleave from the membrane PIP2. It travels through the cytosol to open ligand gated calcium channel in the endoplasmic reticulum. Calcium that have released from endoplasmic reticulum will bind and form calmodulin and do different uh, activity like muscle contractions, neurotransmitter sequencing, etc. Then hydrophobic secondary messengers. What are hydrophobic secondary messengers? These messengers will uh, These are non-polar molecules restricted to plasma membrane or gas that easily diffuse through the lipid bilayer entirely. This where uh, as shown here, there are two examples, that is DAC and nitric oxides. DAC, diacylglycerol. When PIP2 is cleaved, IP3 and DAC are activated or formed, but the DAC remain trapped in the plasma membrane. It slightly laterally to recruit and activate TKC alongside calcium. Then the nitric oxide. It highly diffusible lipid-soluble gas that easily cross cell membrane to affect neighboring cell paracrine signaling, directly activating internal enzyme like guanyl cyclase. As you shown in the uh, as you shown before about the nitric oxide uh, functions, I have said in GPCR in the third class maybe. Then talk about the part 2 topic, desensitization. What is the meaning of desensitization? The desensitization meaning the continuously activated receptor will create some problem. So the activated receptor would be removed or repair so that it will not create uh, continuously activated form. Like what will be danger? Continuously signaling would be break, lead to pathological state like heart failure, several drug tolerance, and cell death. But how should we protect the cell? The physiological process where a cell actively decreases its sensitivity to prolonged signaling, the cell appeared molecular break to physical uncouple the receptor from downstream G protein along recalibrations. What we'll do here? We'll repair the receptors so that it will not activate or continually activate the GPCR or G protein molecules for continuously having signaling. Homologous desensitization silence active receptor. Here, at first, recognition occur. An antagonist bind to active GPCR, exposing a specific intracellular domain. Second, phosphorylation, where GRK, G protein coupled receptor kinase, add phosphorylation to the serine tyrosine residue on the receptor intracellular tail. What would be the result? The newly added phosphate act as a homing beacon through which the inhibitory protein attack and block for the G protein interactions. Then what about heterozygous desensitization? In heterozygous desensitization, a particular pathway will lead to desensitization of multiple different receptors regardless of whether they are actively bound an antagonist or not. The secondary dependent kinase like pKa or pKc act on it, like as here given in the table, homologous breaking or heterologous breaking target only antagonist is bound but multiple receptor active or inactive. Kinase GRK, but in heterozygous pKa or pKc, scope highly specified, here broad. Beta arrestin. What beta arrestin do? During the desensitization, when the GPCR is actively showing that it's active, and ER and then the GRK uh, add phosphate in the C terminal. After that, the beta arrestin come and bind to it. After binding it, it create uh, it uh, it create Catherine-like molecules or called adapter protein that recruit Catherine and AP2 and trigger endocytosis, pulling the receptor out of the plasma membrane and into the intracellular endosomes. Internalization receptors are either recycled back to the membrane, resensitizations, or sent to the lysosome for destruction. Then what is the summary here? When ligand bind to GPCR, it so continuously activate G protein or activate secondary messenger, GRK or PRK phosphorylations that cause beta-arrestin to bind and recruit adapter proteins, and that adapter proteins that is shown here, Catherine and AP2 bind here and endocytosis to repair the receptor. Other for secondary messengers and etc. are given here. You should read it and understand all the topic here. I made some page, I think the summary page, handwritten notes, handwritten messenger. So I'll bring in next class and show that one. So thank you for the classes. Okay, bye.